our company's introduction. So we are the startup originated from University of Tokyo and Riken Institute. So obviously we stationed in Japan, and we specialize in the AI-driven spatial analysis for mainly preclinical drug discovery research. And our mission is to support and accelerate drug discovery by providing innovative whole organ 3D imaging and the data analysis support. And obviously, this technology also can be applied for organoids or spheroids. So hopefully, the audience of today can get some takeaway from our presentation. So today, presentation will be conducted by myself and Professor Etsuo. He is a leading medical scientist in this industry and one of the actually key contributor to the development of the cubic technology. So feel free to ask any questions about the cubic technology itself. And then here's today's agenda. First, we will introduce the fundamentals of the cubic technology. And nextly, we walk through a little bit about our service and products. And finally, we will showcase several use cases and application examples related to pharmaceutical research. So let's begin with looking at the conventional methods used in preclinical tissue analysis. I assume most of the audience are, experiencing, this conventional method. So originally, after finding the drug candidate, obviously we inject to the animals, make the slice and samples, and observe the tissues in 2Ds. So, but however, there's a process has several challenges. So obviously, since we see in 2Ds, there is a lack of the complete organ view, and it's kind of hard to get enough information from the sections, right? So it's time and energy consuming to make the slices every day, but it's kind of hard to get enough information to conduct, right decision making to accelerate the drug discovery. So here's our approach. Basically, cubic technology can offer the 3D imaging at the cellular level. And then here's an example of the mice brain with the blood vessel and cancer tissues. And then, of course, not only providing those images, but also we can provide the data set. It can be used for PK, PD, or safety testing to support the R&D activity of the pharmaceutical industries. And then, so how does it work? So we're going to briefly touch on the technology and science itself. So our platform integrates basically 4 core steps. One is the tissue clearing, and the 3D immunostaining, and the imaging, and finally data analysis. This combination allows us to perform highly detailed and comprehensive 3D analysis across entire organs. And then the first steps begin with the organ extraction and followed by the chemical clearing and the uniform staining. And then actually we conduct the scanning by using the light sheet microscopy. And then we conduct deep learning based analysis. So here's what is unique in the tissue clearing. So in order to see inside the body, if there is a color, obviously we cannot see inside. So firstly, we are trying to remove color and fat out from the organs, which is called delipidation. And then following, and then after that, we adjust the speed of the light so that we can see, we can clear equally of the organs. And then not only make it transparent, in parallel, we are staining our staining technology ensures the uniform dye penetration across the entire samples. For traditional staining, we can stain only surface of the organ, but our technology can allow the antibody penetrate into the deeper side of the samples, so that we can achieve uniform staining of the the target. So our work has been featured in numerous high-impact journals, including Nature, Cells, and Science. We believe this reflects not only the scientific credibility, but also the broad application of our platform, including the organoids and spheroids. So with that technical foundation, let's now move on to the second part of today's session, our service and the products. At the CUBICStars, we offer the preclinical support service that combine 3D imaging and AI-powered analysis. So firstly, we receive the post-drug administration samples from pharma or research institute, and we conduct the 3D imaging, including the clearing and staining and imaging. And also we analyze those data. So we conduct image analysis and the statistical analysis. And then finally, we provide the PK and PD tox reporting, which can be directly used for the drug evaluation. So here's workflow. So we receive the fixed sample from our clients. We conduct those, clearing, staining, scanning, and analysis, those process, and then finally send the data back to our clients. Normally, it will take 2 to 3 months at shortest, depending on the, how challenging it is. And then we are accepting, developing the protocol itself. And then the CUBIC platform enables various applications, including PK, PD, and safety pathology. For example, we can quantify drug localization in brain and evaluate tox effects in kidneys, and also analyze the immune cell response in the brain. So these insights are difficult to achieve with the conventional 2D histology. And beyond imaging, we also provide a full analytical pipeline. So our platform supports data visualization and also spatial location information, and also the statistical quantification. So we identify, how many cells or how many signals and where it is, and we can compare between the group of the samples. And we also provide the reagent kits, which is optimized for the CUBIC workflow. These are manufactured under the strict quality control at our factory. And you can see our website for the details, but we offer the reagent kits which can be used for the tissue clearing process, and also tissue staining, 3D staining process. And also we offer reagent kits which can be used for the organoids and spheroids. So if you have interest, please see the details about products on our website. And those technologies already on the market and being adopted by top-tier institutions across the globe. So this collaboration reflects the adaptability and the credibility of our platform. Okay, then finally, we will show some of the practical examples of how CUBIC has been applied in the real research context, including the CNS disorder, tumors, and organoids. And I will hand it over to Etsuo, and he will share his work in academia.
Okay. All right, thank you, Yoji. And hello, everyone. I am Etsuo Susaki. I'm a medical researcher based on the Juntendo University, Tokyo. And also, I am an executive officer at the CUBICStars, particularly being in charge of R&D activities, the technical instructions, and the support for users. So today in my part, I will introduce some use cases of our techniques, particularly in the biomedical field and drug discovery. So in the first case, I would like to introduce the analysis of Alzheimer's disease models. And in the first slide, we can see the whole brain immunostaining and the imaging analysis results of amyloid beta expressing brains. So of course, in this field, the analysis of the distribution of such A beta deposit or tau deposit is so important. And our techniques enabled the complete homogeneous staining of amyloid beta, as well as some structural markers like Bessel, the smooth muscle actin. And then this data can be used for the subsequent quantitative analysis. And the other important and the good thing to use such 3D imaging technology is that these technologies is very good at identifying the rare biochemical and cellular activities inside the whole entire organs. So in this case, the researchers identify the neurons that are engulfed by the activated microglia. And so this is a very rare phenomenon, and but this 3D imaging can provide such the information, and also not only identifying these phenomena, but also that this data can be used for the quantitatively analyze the numbers and distributions of such specific phenomenon inside the brain or other organs. And here is another case of whole brain imaging and the analysis of the tau plaque pathology. So this work recently published in the Brain Communications. So if you're interested in details, please check out these papers. But briefly, I would like to introduce the content. So in this case, the researcher successfully stained the whole brain with tau antibody. And that's a very famous tau antibody named 88. But the 88 is also known as one of the very difficult antibody to stain the 3D volume tissues such as the whole brain. So our ATV techniques can only enable such a whole brain staining and imaging of this the tau plaque across the sample. So not only visualizing the distributions, but also the researcher successfully calculate the whole distributions of tau plaques across the brain according to the brain regions. And not only just calculate the whole distributions, but also they tried more intensive analysis to evaluate the working hypothesis in this field, such as the tau plaque is distributing according to the neural circuitry. So to evaluate, to prove this hypothesis, they try to calculate the density of tau deposit across the brain regions, and then identify the gradient of the tau deposit at the specific brain regions. And then they successfully demonstrated that this gradient is consistent with the neural circuitry. So that's why the analysis proved that this tau deposit distribution is consistent with the working hypothesis, that the tau plaque is distributing or delivered across the anatomical network of the brain and neural structures. And in this slide, I'll show you different applications in the drug developmental field. So in this case, researchers made CDX, the cell-derived cancer xenograft model in the mouse, and then the mouse was administrated by a specific anti-cancer drug named trastuzumab. That's the antibody drug for a HER2 molecule. And then the researchers collected the sample and took the image, the 3D image, to evaluate the distribution of trastuzumab inside the whole mass of whole tumor mass. And as you can see here, surprisingly, the distribution of this drug was completely inhomogeneous inside the tumor. So that suggests the tumor microenvironment may affect the efficiency of drug delivery and the penetration at each position of the tumor. So this analysis also paves the way to the understanding how the tumor microenvironment is controlled, or how the microenvironment is can affect the drug distributions, as well as this analysis can open the door for the new drug discoveries related to the basic structures and the functions according to in terms of the drug delivery inside the tumor mass. So here's a movie of this whole tumor mass. So the data was acquired for the CD31 as a vascular structures, and also the trastuzumab, the drugs. So as you can see, the whole tumor mass was successfully collected and evaluated. And other example, particularly for the drug discovery, is the analysis of the cell activities inside the organs. So in this case, the researcher focused on the drugs for the central nervous system. And after drug administration to the mouse, they collected the whole mouse brain and then stained with anti-CFOS antibody to see the neural activities across the brain. So after staining, each CFOS signals can be identified as a coordinate, and then these coordinates can be counted or quantitatively analyzed according to the atlas regions. So our software can provide such analytic platform to compare the different experimental conditions in terms of the neural activities. So here's one of the simple description of the neural activity by using the bar graphs, but also our software can provide whole brain heat map descriptions according to the neural activity. So in this case, the heat map is related to the number of cells in each brain regions. But when researchers collect the sufficient number of cells for the subsequent statistical analysis, our software can provide the analysis results, such as the p-value comparisons across the different experimental conditions. So such analysis also provides the platform to identify and analyze the drug effect inside the whole brain, as well as the other organs. So I just show you a very limited number of application studies by using our techniques. So, but the combination is numerous. So we can consider several modalities, drug modalities, diseases, and compatible organs, and the compatible molecular targets. And all the combination can be available by using our techniques. So we hope the new researchers can come up with the new good applications by making use of our techniques, and they're really happy and appreciate if you use our techniques for your future research. So as a take-home message, I just show you the summary. So our cubic 3D imaging is a groundbreaking techniques and for the spatial analysis of animal organs, as well as the organoids and spheroids. And also now the cubic techniques can serve as a fundamental platform for various research fields. So if you're interested in using our techniques, please feel free to reach out to us, and we are very happy to help you introducing our techniques and use it. So thank you very much for taking the time, and we are really happy to take any questions.
Thank you very much, Yoji and Etsuo. It was a really, really, really wonderful presentation, very interesting. I'm gonna unmute the attendees. So now you're all supposed to be able to unmute yourselves in case someone has a question. You can also use the chat. We will give you like a few seconds. In the meantime, I do have a few questions. Oh, Jens, go ahead.
No, okay, no, it's fine. First of all, my apologies for being late. Sorry for that. Very wonderful presentation. Thanks a lot. I really like your way to quantify nuclear staining in the organoids that you have shown. Would your method also allow you to quantify really in absolute numbers the occurrence of more complex cells, like neurons, for example, which are not just round but have a complex morphology?
So in our cases, so we basically stain the whole nuclei rather than the soma. So and even in the complicated cells with the complicated structures, basically the counting can be completed by using the position of nuclei. So that's why, yeah, we believe it would be suitable.
But you will still count the nuclei, right? You will not count the other morphology of the cell.
So of course, that's totally dependent on the experimental design. So if you are interested in the specific cell morphology, so you can stain the like the surface of the cell membranes and then calculate the morphology of cells. But in this case, I guess some sparse labeling techniques will be required because yeah, the segmentation of all cells, all stained cells, are a little bit tricky. So yeah, but depending on the experimental design.
Thanks a lot.
Thank you. Any other questions? If not, I will go ahead with my questions. I have a question just out of curiosity. What is the biggest tissue that you have stained, that you have cleared and stained and imaged successfully? Because it's really interesting, the clearing method, and especially this penetration enhancer that I had never heard about, because I know that there are different clearing techniques, but one of the issues was then the antibody penetration. And I think it's great that you have this reagent that allows for an optimized antibody penetration. So I guess this allows for staining of relatively big tissues. So what do you think is the biggest tissue you have stained?
So as for our service products based services, that we've already provided the staining of whole rat brain or whole rat organ. So that's usually can be stained with our techniques. At least in the research level, where we tried the whole marmoset infant body, that's almost close to the size of the adult mouse, the centimeter in length, and that's successfully stained with the nuclei dyes, and then acquire the images. So that's a maximum size we tried. But not only our techniques, but also other researchers tried, the perfusion techniques to stain the entire sample. So our techniques is based on the simple penetration, the simple immersion and the penetration. So that's why it's very easy to be implemented and used. But if you consider such a really advanced technique, so it can also be used with our techniques.
That's really cool. It's pretty big, 1 centimeter.
Yeah.
Then in slide 21, you were presenting this image of the microglia engulfing the neural cells in the AD model. And that was very interesting. I wonder, because you can see very well the microglia morphology and everything. So I wonder what is the highest resolution you have done for a study. Can you go to intracellular organelles or something like this?
So it's also dependent on the design of the experiment. So for example, in this slide, so I show you the low resolution images with amyloid beta staining. So in this case, we used the 1x objective length. So the resolution is something like 10 micrometer cube voxels, or something like that. But in this case, we use a 10x objective. So that's why you see the effect of the tiling, the stitching, and then you can see the more detailed cellular morphology inside the images. So we can provide such kind of a little bit higher resolution imaging. But the one issues we adapt such a high resolution imaging, the data size is also increased. So that's why that's also totally dependent on the experimental designs, and we recommend to get the images with minimum resolution you require for your experiments.
That's really interesting. So potentially, if you have a project about a drug distribution in brain, you can first check the distribution of the drug in the whole brain, and then go with the higher magnification in the cellular and subcellular level, right?
Exactly. So in this case, so we first use low resolution microscopy, and then we focus on the specific regions by using the higher resolution microscopy. So not only the large systems, when you cut out the specific regions, so you can also apply the confocal microscopies or two-photon microscopies to see the subcellular resolution structures and the distributions. Yeah.
It's really, really interesting.
I have a question around at the same slide, actually. Can you see there also protein aggregates like in the Alzheimer model plaques?
Well, so this model actually shows the protein aggregates of amyloid beta. So this antibody identifies the plaques of the cell aggregates of the amyloid beta. So that's why the deposit you can see here is basically related to the deposit and aggregates of this protein.
It looks very uniform, right? Wouldn't you expect a bit more like a I don't know, asymmetric distribution?
Oh, yeah. So in this case, this is like 9 months old, and so it's completely progressed. The disease is completely progressed. So if we younger animals, the distribution is more sparse, and as you say, it's non-homogeneous. And so in this case, this is a tau model, newly reported in this paper. So in this case, you can see more inhomogeneous distributions, and that's totally related to the neural network that support the tau deposit movement from the origin to the peripheral regions of the neural network.
Okay, yeah, that makes sense. Thank you. You haven't done something like that for a Parkinson model, right? Where you see Lewy bodies. Or did you?
Oh,
Can
no.
you hear me?
Sorry, I missed you.
No worries. My
Yeah,
question was whether you did something similar also for a Parkinson model where you would see
okay,
Lewy bodies.
I see. Yeah. So at least for the research project-based experiments. So we tried the staining for anti-synuclein or something that's more related to the Parkinson model. And our techniques also successfully stain the whole brain distribution of the alpha-synuclein deposit. So yeah, it can also be applied for the Parkinson studies as well.
Thank you.
Yes.
So I have yet more questions if no one else wants to ask. Just out of curiosity, when you presented this trastuzumab drug, the slide 25, it was really, I think it's really cool that you can see this homogeneous distribution of the drug. Did you then follow up in this project and see if this led to, in different time points or something, to the tumor growth or like any cellular consequences of the treatment?
Yeah, so actually this is just a one time point experiment. So we just corrected the one samples and evaluate. So yeah, it's really surprising results. We didn't expect such a result, the inhomogeneous distribution results, but actually the tumor has a really complicated microenvironment, and that's why the distribution is also inhomogeneous. So the potential project would be, why and how this drug distribution can be determined across, at each microenvironment. So when researchers successfully identify such mechanisms, that also opens the possibility to modulate such drug distribution efficiency inside the tumor mass. So this result clearly shows that you should consider how the drug is distributed inside across the tumor mass. So some area is not so efficiently treated in these cases. So that's why the modulating and improving the drug distribution will be very important for future cancer studies and the treatment.
Yes, so as I said in the beginning, for those that couldn't connect on time, this meeting is recorded and will be available in the OrganoTherapeutics channels. So basically our website and LinkedIn for viewing afterwards. It will be edited and then it will be uploaded. So feel free to check it out afterwards. We were very, very glad to have you, Yoji Otsuki and Etsuo Susaki from CUBICStars. Do you have any further comments also from your side?
No, not specifically, but thank you for attending today's webinar. And we believe most of the people stay watching our, up to the end of the Q&A session. So really appreciate your interest on our technology. And again, thank you for today's opportunity.
I just want to have final words to also thank you for being here. It was great to see you again. And well, looking forward to future interactions.
Yes. Thank you.
Thank you very much, everyone. As we said, we keep open to feedback and questions. Have a nice rest of the day. For the people in Japan, have a nice evening, I guess. And yeah, thank you very much again.
Thank you so much. Have a great
See you. You too.
Thank you.
Thank you very much.
Bye.